we understand the main reaction which gives us ammonia, this is deamination. So we have a lot of amino acids in our body. We need to catalyze it, to degrade it, and in the end of the degradation, we will have ammonia. What else we can deaminate in our organism? I remind you, biogenic amides will be deaminated, and nucleotides also will be deaminated. Next topic, we will talk about nucleotides. So adenosine deaminase is a very active enzyme. It involves the immune system. So let's underline again, revise. Deamination of amino acids will give us ammonia. Deamination of biogenic amides will give us ammonia. Deamination of nucleotides will give us ammonia. And last, fourth way of ammonia getting, it will be putrefaction in gut, in digestional tract. If we will not absorb all amino acids, some amino acids will be putrefied by our bacteria, and we can get ammonia in the end. So what we will do with ammonia? We can't transfer ammonia in free form. So that's why we will transfer ammonia in form of amino acids.
طيب واحد ما فينا نحول أمونيا الآخر هو جنبك أنت عايز إيه تاني؟ حول طبيعي
So, ammonia in brain. In our brain, we have a lot of neurotransmitters. So, reaction of deamination in brain providing very fast velocity, and that's why we can get a lot of ammonia in our brain. How we will transfer it? So first idea, we can connect it with alpha-ketoglutarate again, and we will form glutamate. Alpha-ketoglutarate will be taken from Krebs cycle. If concentration of ammonia is very high, we can add one more ammonia molecular to glutamate, one more ammonia, and formate glutamine. Glutamine, this is amide of glutamate. I remind you formula of glutamine. So let's count. One amino group, second amino group. So transport way for ammonia from brain, usually this is a glutamate or glutamine. If we have very high concentration of ammonia, one more way, we will use oxaloacetate. Again from Krebs cycle, and get aspartate. Same reaction when we add one more ammonia will give us asparagine. But we will prefer this way because when we convert glutamate into glutamine, we spend only one ATP. And if we will convert aspartate to asparagine, we will spend 2 molecules of ATP. That's why our organism prefer to transfer ammonia in form of glutamate, glutamine. But it allows to transfer it also in form of aspartate and asparagine, but we will spend more energy. It's happened in case of very high concentration of ammonia. The main idea why it's happened in that way, that rain contains specific enzyme, we already know it, glutamate dehydrogenase. So glutamate dehydrogenase. But muscles, almost absent in muscles, almost absent this enzyme. So muscles need to have another way of ammonia transfer. So, for example, what we will do with muscles? In muscles, we will get alanine. Do you remember reaction of alanine transamination? So this is in reverse way. So pyruvate convert into alanine in muscles. After it, we transfer alanine into liver. Liver will convert it again into the pyruvate. Pyruvate can be transferred in free form, but usually liver will use pyruvate for glucose formation. That's why in muscles we have a specific cycle. It's called glucose alanine cycle. One more time. In muscles, we will convert glucose into pyruvate. Pyruvate into alanine. Alanine will transfer into the liver. In liver, all steps in reverse way. So alanine convert to pyruvate. In reaction of gluconeogenesis, pyruvate will convert into the glucose, and we return to muscles. That's why it's called cycle. It's very brief information, so you need to find this reaction in books. You need to find this cycle in books. But you must understand that we will never transfer ammonia in free form, because ammonia is toxic for us. It will change our pH of blood. It will lead alkalosis. That's why it's impossible to transfer ammonia in free form. Yes, of course, small amount of ammonia we can transfer in free form, but usually we need to form a transport amino acids. What's happening in our liver? Only liver will convert ammonia into urea. So this is a prerogative of liver. No any other organ will do it. So in hepatocytes, here please close. Yours, maybe we will open the door.
Close this one.
Give it 10 minutes. Okay. 10 minutes. 10 minutes and you will be free. In liver, in hepatocytes, we will start our reaction in mitochondria. I like only names, but I ask you at home, write formulas of these chemical compounds too. And we learn urea cycle just like we learned Krebs cycle by formulas. So, in liver, we will take ammonia connected with carbon dioxide. These steps present in mitochondria, and we will form carbonyl phosphate. Carbonyl phosphate. Enzyme name carbonyl phosphate synthetase. I will write short. Carbonyl pH phosphate synthetase. We spent 2 molecules of ATP. Again, we spend energy, we spend energy, we spend energy. So formation of urea, this is a reaction when we spend a lot of energy for detoxification of ammonia. But we need to do it in other ways, we will poison by ammonia. So carbamyl phosphate connect with specific amino acid which name ornithine. That's why urea cycle also named ornithine cycle. Ornithine. Ornithine is non-proteinogenic amino acid. That's why we will never use it for protein synthesis. It's involved in urea cycle, and also from ornithine we can get specific biogenic amine, spermine and spermidine. As a name, you understand that it will help our cells divide because it will discover in sperm. So ornithine is non-proteinogenic amino acid connect with carbamyl phosphate. We release phosphate group, so phosphor will go out, and we can get another non-proteinogenic amino acid named citrulline. Citrulline. So citrulline will be go out from mitochondria by helping of specific transport protein. So in mitochondria we have 2 membranes. So it's very difficult to penetrate this membrane. That's why we need to put some transport protein. Citrulline in cytosol connect with aspartate. And we know that aspartate also transport form of ammonia. That's why 1 ammonia molecular we can take in free form, and 1 ammonia we can take from aspartate. No, it was not ammonia, it was amino group, NH2. So citrulline with aspartate convert into argininosuccinate. It's difficult names, but when you will see the formula, it will be very similar. You rewrite citrulline and aspartate and call this huge formula named argininosuccinate. Argininosuccinate. So argininosuccinate synthetase. Argininosuccinate, synthetase. And again, synthetic reaction by synthetase catalyzed by helping of ATP energy. And here we will spend ATP into AMP molecule. So we will spend 1 molecule, but 2 high energetic bonds. So it's equal to ATP. So again, for our organism, it costs much in high expensive reactions. But we spend this energy to detoxify ammonia. Argininosuccinate, by helping of argininosuccinate lease, so this side, I write that argininosuccinate synthetase, argininosuccinate lyase. We will cut fumarate and get arginine. Now we already know arginine. Arginine is normal, our amino acid that we use for formation of protein. In childhood, arginine is Semi-essential, what does it mean? That amount, what we synthesize in urea cycle, not enough for us for protein formation. That's why in childhood we need take arginine as a food source with meat and some other elements. But in adult organism, arginine can reduce from later cycle. But again, if we are healthy persons, so proteinogenic, semi-essential amino acid. Arginine will convert to ornithine. And we cut urea. And look at here, in urea, we will excrete two nitrogen atoms. Urea is non-toxic chemical compound. Urea provides osmotic pressure of our blood. So urea is useful for us. But we have a lot of urea and we will excrete it in urine. So urea is normal component of our urine. And what we will do, we will return ornithine into mitochondria. So do you remember? In my case, it's wrong destination, but we will use ornithine again in formation of citrulline. So that's why it's called cycle. Arginine degraded by helping of arginase. Arginase. Urea from liver will transfer into blood, filtrate by kidney, and we will excrete it by urine. Increase of urea concentration in blood show us kidney failure. All time students make a mistake that when I ask you what organ will suffer if urea concentration is high, almost all say
Kidney.
liver.
No kidney.
But now you understand it's kidney. Urea synthesized by liver and filtrated by kidney and excreted by urine. If we don't have kidney, urea concentration in blood will increase. So we use counting of urea amount by diagnostic of kidney diseases. At home, you will again read first topic, read second topic. I'll check all your written tasks. Next class, I'll give you your 2 marks. I remind you that on Monday at 6:30 p.m., I can ask your extra work. It will not only your group. It will Russian group, general benefit for education on medicine.